position. Is this one on? Oh, it's there already. Oh, yeah. This one, yes. And this one. We're going
Okay,
to
everybody,
go
now we're
for,
getting the final mention made. I'd like to welcome you all to the second session of today. My name is Ian. I'm the chair for this session. I'd like to welcome the members of the jury, David Garcia, Trinidad, and Fatima. I'd like to welcome the speakers, Robert, Oluwasegun, and Samuel and Manuel. This promises to be a very enjoyable session. Quite a diverse range of topics to be presented. You give the presenters the very best of your attention, and we'll have time for questions at the end. 15 minutes for the presentation, 15 minutes for questions, and off we go. Very best. I'll give the speakers 5 minutes warning, which is 5 minutes. I start now. Yes, if you like. Thank you.
Okay. 5, yeah?
Yeah?
So. Good morning, everybody. My name is Robert, and I'm going to present to you temporal and spatial variation in adult and chick diet of the sandwich terns in the southern part of the North Ocean. So, what are the sandwich terns? These are very elegant birds that are characterized by a signature beak, which is long and black with a yellow tip, and short, stubby legs that are also black. This differentiates them from different tern species. They're known as a plunge diving birds, which means that they plunge to catch their prey up to 2 meters, and they're particular specialists. That means their primarily feed is fish. Here you can see the global distribution, but the most prominent colonies can be found in Northern and Western Europe and the Caribbean, where most of the studies have been done as well. From the literature, we could find that they eat a variety of different prey species, depending on the availability of the field and other effects like climate change and similar. Mostly it's the members of these fish families, most prominently clupids and ammonites or sand eels. And the second most prominent is the nereis or the ragworms. Regarding their breeding, so they are colonial birds that form large colonies in the sandy beaches, sandy barriers, barrier islands, and in the Caribbean coral reefs and coral islands. They form large families and they can number over a couple thousand pairs. As well, the time period for the breeding period, it depends on where in the world, but usually from end of May and to the middle of July. So regarding dietary studies, so dietary studies are important as especially for the bird species, as most of the diet studies have been done solely for the chicks. It's very easy to do it, and if you find a colony, it's pretty much go. While for adult birds, it's very hard to do it as they spend most of their time feeding at the sea. So a new way should have been developed to study this diet. So here where you can see are the most prominent dietary studies for the adults of the sandwich tern species, primarily in Germany, Netherlands, and North Carolina and Puerto Rico in the Americas. So the past research practices were primarily done using the stomachs and regurgitation, which was very intrusive and invasive to the birds, and there was a lot of loss in population. But in the recent modern times, they switched to the more easier, more cheaper, and non-intrusive method, which is the fecal analysis. As I mentioned before, the adult diet is poorly studied, and that's why we did this. Now, my research questions. Research questions are going to incorporate the degree of interseasonal variation in the species composition and prey length for the adults, the same for the chicks, and then compare the two to see is there a prominent difference between the two. And my research goals will be to perform the temporal variation to go beyond the limitation of the previous studies, which were usually concentrated in only one very, very short period and over only 1 or 2 colonies, and provide more information about the tern diet composition for the adults for future studies, fisheries, and their conservation, as they are in some places of the world registered by IUCN as vulnerable. Now, materials and methods. Here you can see the 3 colonies that I've studied. Two are found in the Netherlands. One is called Haringbillet, the other one is Grind, and one in Germany that is Norderuk. The all three colonies are very famous in the marine biology regarding bird studies. Materials and methods. So regarding materials and methods, my material was the fecal matter that was collected from the colonies directly. The method of collection was the use of the trays and the observation by professionals for the chicks, which I'm going to explain a bit later to you. Samples have been collected for the 3 colonies in the 5-year period from 2016 to 2021. And as well, besides them, the reference fish were collected to compare the sizes of the prey to the one found in birds. Treatment and proceedings. So the collected material was taken to the lab, it was filtered, cleaned, separated, and sorted. The sorting has been done by species or group, depending how far we went to the identification of the breed, and put in the epitope tubes. And then identification was done by stereo microscope and using the J, where we identified the otoliths. Here you can see a simple step-by-step process of how it went. So from the tray, collection, sorting, what I see on the microscope, identification, and plus the reference fish. For the chicks, it was a bit different, as the chicks should not be disturbed, and it has been proven that they can affect negatively their growth if they're often disturbed. The sampling period was in shorter 3 period, only in the Haringbillet colony, as that's the most famous and largest colony, so they only did it there. The collection was done by professional observers from a bird hut that is found next to the chick to the colonies, and they recorded all the prey species and similar on the sheets and photos. The treatment, they gathered all this, and then they calculated the prey's length based on the bill length, which has been given by my supervisor Walter Curtin's previous papers. So now a bit visually to interest you a bit. So how do we identify the otoliths? Primarily the shape. Every fish has a different shaped otolith, and that's how we can distinguish species in most cases. After that, we measure the size of the vertical length of which we transformed later into an appropriate number that will correspond to the size of the fish, as otoliths grow with the fish. So the larger the otoliths, the older the fish, or the larger the fish. And lastly, the age group was done to be converted. Here are the 2 formulas. The first formula is to transform the width of the otolith to the appropriate size, and then correspond to the appropriate fish species for both adult and chick. And then we compare them to the otoliths. This is the formula for the bill length in series. Okay, regarding statistical analysis, I performed the ANOSIM and the MS, Kruskal-Wallis, plus Dune test, and chi-square. ANOSIM has been used because it's very good for population compositions, and the MS to represent that more visually, to see is there similarities or dissimilarities. Kruskal-Wallis has been done to compare the different years, dates, weeks, and chi-square test to compare different fish lengths between the colonies. Now regarding results. So in this study, there was almost 50,000 otoliths that were extracted from the bird feces, which I personally did almost 9,000 of them myself. After that, after performing a certain statistical analysis, like ANOSIM, we found that there was a significant difference between the different groups in both week and year, and that the most prominent prey species were sandhills and clupets, which matched the previous literature. Here, you can see the graph with the temporal proportion between the different years, going for 2016, 17, 18, 21. And you can see the proportions of sandhills and clupets. As you can see here, it's pretty much balanced, except in the last year of the 2019. And that was for the Grind colony. For Norderuk colony, it was a bit different. As you can see here, clupets were far more dominant, especially in the beginning of the season and the end of the season compared to the sandhills. The most sampled colony was the Haringbillet, which was done on many occasions. And from what you can see here, it varies naturally. So primarily there was the sandhills that were dominant at the beginning, then the clupets took over, and then by the end of the season, other prey like ragworms overtook. And you can notice this trend slowly, except in 2021, where the ragworms were not utilized almost at all. Here, just to show you simply, regarding the age class specification of the most prominent species, like sandhills and clupets, we had a couple of different age classifications. They were separated into the adult sand eels, adult clupets, juveniles 1 and 2, larval and post-larval. And from here, you can see that the most chosen size of the bird or age was the post-larval clupets and the adult sand eels over all colonies. Regarding statistical analysis, so age class proportions of sand eels and clupets were compared together between the three colonies in different decades and by the same year. So what we found between decades, there was no significant difference between them. Between 2016 and 18, which were the only 2 years where all three colonies were sampled together, so I only compared between the three. 2016 was significant, while 2018 wasn't, and sand eels showed significant difference between three colonies in their size and numbers compared to the clupets. As well, individual colonies of northern herringlet were significant by the same proportions compared to the grid. Now we go to temporal variation. So what is temporal variation? So temporal variation is to show how the diet changed through the sampling period. So as you can see in the beginning, I will help you to understand this a bit easier. So as you can see, for example, juvenile type 1 sand eel, the first year of 2016 for the Benin colony, showed a decreasing trend by, it was selected most prominently in the beginning and then started decreasing, while for example, in Norderug, for the larval clupet, they were selected less at the middle of the season, especially here, and then highly later. For Herringville colony, it was because it was through through much more years, you can see more trends, and the trend is quite easy to explain. Sand eel was decreasing, clupets were increasing, and similar. This was done for multiple years, so you can see how the trend leaves. Regarding fish lengths, we found there was no significant differences between the fish sizes between the three colonies in 2016-18, so comparing the three. As you can see, the sizes are more or less the same in all three colonies. Same was done for the sand eels. In 2018, it was the similar trend. As you can see, all the sizes for appropriate classes were like that. In 2019, it was a bit different. As you can see that in Norderug, the sand eels were larger, much more compared to the herringlet, as it went from 12, most on average in the herringlet, and in the Norderug, it went from 14 and up. And most interesting years, 2017 and 21 showed significant change. As you can see, the variation of the adult sand eel from like very large to very small to very large, and significant decrease in the size of juvenile 2 herring from the beginning to later point. Here you can see the analysis. Results for the chicks. So for the chicks, the same was done, and you can see the temporal variation. As you can see, for chicks, sand eels were more prominent at the beginning, clupets later, well, very similar to the adults, what was done for herringlet colony. Temporal variation as well, so you can see the decreasing trends. So adult sand eel was more prominent at the beginning, while later the large species like herring, age 1 and similar, started to increase. And same happened for the old sampling years. This is a statistic, so it shows for the 3 years that there was a significant difference between them. Fish lengths here, it was a bit different because, as you can see, as the chicks grow, the fish size grew with them. So that means that the smaller the chick, the smaller the fish, and as they grow, the fish increased. As you can see from the example juvenile type 2 herring, you can see it slowly increasing. For sand eels, it was the same. As you can see, adult sand eels started increasing in size growth to chick as the season went. When comparing the two, you can see that chicks were fed much more with sand eel over the compared years compared to the adults, and adults ate more the other species. Here you can see this visually. So visually you can see the clustering, and you can see there was no much clustering whatsoever. So there was no many similarities between adult and chick diets. Except, for example, in 2000, in the first graph, you can see that, for example, the week 4 was quite similar, but this was not a common trend. Here you can see the analysis, so they were quite different but similar. Here is the difference in the size, so you can see that the adults ate mostly fish, which was, for example, adult sand eel, between 12 and 14, while the chicks ate between 12 and 16. So chicks were eating more. Regarding the discussion, so what are the limitations of my methods? So this new method which we used was not published before, and it was very effective. The feces are very useful, cheap, and can be performed by almost anybody if trained properly. Other methods were more invasive and more expensive. Otoliths, the otolith size, the only negative thing regarding my method is that very small otoliths, like under 300 microns, can be lost. But some studies have shown that this is actually false, that in pectivorous birds, especially marine birds, you can lose the otoliths very rarely as they pass very fast through the gastric tract. Regarding this, so here you can see that in this study, the three main group, you can see what we found in this study and what was compared to the previous literature, and so did it match or not match? The three major groups matched. The dominant clupid species of sprat and herring matched. The sandeel were not identified to species level because this was impossible from the otoliths. There was no distinct differences in the green proportions that was matched. The only difference was in the Harik and the Norderug, where the sandeels were usually dominant, but now were clupids. And the clupids were dominant. Now for temporal variation and prey, we can explain here that so chicks were given bigger food because this matches their biology and seasonality. Some fish that were found later on in season in bigger numbers than usual can be acclimated to change in the warmth of the oceans. Large clupids and sandeels were dominant prey species. This matches the previous studies that were done, and the chick and the fish size went from adults eating larger fish to smaller fish because they try to contribute to their young, which matches the optimal shearing theory. And for the future recommendations, for the adults, we propose multiple continuous sampling, mirror the previous studies, and do both intensive and extensive sampling every 3 to 4 days. While for chicks, continuous sampling observation should be done by professionals in good weather conditions and from a decently separated hut, and everything should be recorded to avoid for mismatching and under or overestimation, as well as after the standard protocol. And by last, I would like to say thanks to my supervisor, Walter Bigren, for all the support he provided me. And here you can see all the photos that were done. Everything is referenced. All the references for the introduction, for the materials, and discussion. Thank you for your attention.
Very much, Robert. I'll invite questions from members of the jury first. I'd like to go first.
I'll start. First of all, thank you for your presentation. It's a lot of information to digest in a short time, and it's a lot of work behind all the presentations. And I don't know if I maybe miss it, but what do you think that's the main on the diet, all diet?
So regarding the main differences, so they eat the same species, so it's not about the composition itself, it's about the quantity and the age classes that were selected. As you can see from, do you want me to show again the slides? Okay, so for example here, I'll start with here so we don't lose too much time. So you can see like chicks were fed primarily more with the sandeels because at the beginning of the season, the sandeels are at their adult stage, so end of May, so that's when they are usually by biology their adult stage, so they're fed to the chicks much more, while adults eat them in lesser proportion and they eat less, so they eat smaller sandeels. This matches what I mentioned, the optimal foraging theory, which says that as the birds, particularly birds, even other animals like mammals, this can happen, switch their diet or change their diet preferences as they have young. So in the beginning, they eat larger, so they have energy to care for the young, and as the young grow, they eat less and less because they can't carry this much prey. As in the beginning, I mentioned they are single prey holders, so they cannot carry more than like 2, 3 fish at once. So they can't feed themselves and the young at the same time.
Regarding the method you have, and you explained that this is first time.
Yes, this was not published before. So they did it for this organization, but it was never officially published.
And what do you think that are the weak points of business? Stronger points against all the videos or
Yeah, so the major negative point regarding this methodology is that the fish species that are very small, like for example the sticklebacks and similar, which have very very small outlets, so under 300 micrometers, which is impossible to see by the eye, are lost in the filtering process. That's the major negative point. But we confiscate this by taking the other prey remains, like the stickleback spines or the jaws or from the worms, for example, or the vertebra. And then from there we can say, okay, there was the species, but still we don't know how much and how much. So this needs to develop as the studies go. So the major plus point for this method is it's intrusive. So the birds are not disturbed whatsoever. These birds are known to defecate next to the nests. So basically before the collection, the researchers go, they leave the trays around only 10 to 15 nests, and they go and they come back 3 4 days later when the birds are feeding, and they just collect the trays. So the birds have as least contact as possible, compared to the stomachs and regurgitation, which were quite, let's say, molesting for the birds, because the birds have to be killed to get the stomachs, and regurgitation can leave scars and trauma to the birds. So fecal matters it's cheaper, it's easier to perform, there is no long-lasting negative effects to the birds in population compared to the birds.
Maybe one more to that.
That's the last one. How do you think that, for example, genetics compared to some genetic analysis in there?
Probably, especially for example, as I said, so sand eels are, at least to the current scientific research field, impossible to distinguish by the otolith. So we know it's sand eel, but there is 4 species of sand eel in the North Sea, so we don't know what they are. What they used to do is to sample in the extra otolith sampling, which I mentioned. So they go in the same time as they sample the birds, they sample the fish, which is found in surrounding waters, and they distinguish, okay, the most dominant sand eel, for example, is rayet sand eel, so they presume that they mostly feed on that. But as you said, for example, regarding the genetics, maybe what they can do is try to take some genetic material from the birds when they feed the chicks, for example. It would be a bit more intrusive, but it can give us more valuable data. Or they can maybe try to extract the DNA from the otoliths, if that is possible, and compare it to the DNA from the otoliths taken freshly from the fish, in case the gastric juices did not dissolve it.
The members of the jury.
Okay.
Excellent.
First, congratulations. This is very, for me, it's a very, very nice work. Especially because of the method I'm used, which is non-invasive and your strength is clear, and I think it's something needed. So, first of all, the presentation I like it very much because it's clear, and only problem. Perhaps we went too fast over some slides. I had no time to see them.
I
Your presentation. You may skip those slides. You are not speaking over.
Sure. Primarily, I had a lot of results and I could not exclude them. Then it would make sense to connect them between them and each other.
Yes, I understand.
No. I know that I went a bit fast, but that's why if you want me to explain some of those slides better for you,
Perfect. Very good. But slides and all the chat. And put those extra slides at the end. So you may cover them if there is a question about, but the reader is not expecting something. It's totally minor point because I like and the picture so for me, I don't know what you think, but that's the worst. Are you publishing this?
Hopefully, not my thesis exactly, but I work in the organization called PIPO, or Forestry Service of Flanders, and they are collecting this for the last couple of years, and they hope to publish this all together with my name as one of the helpers. As if you remember from the 50,000 quadrilaterals, I collected almost half myself.
Well, that publication published in the methodology, I suppose is the novel results, the method itself, being non-invasive, but beyond the new method, what do you think, what would you highlight as the main problem beyond that methodology of your discovery?
So, as I mentioned before, most seabirds, especially smaller ones, which usually spend their time feeding in the sea, only chick diet is known, or what adults eat when they have chicks. It's not actually known what adults eat when there is no chicks present in front of them. So there were only 4 other studies performed before, as on the presentation. So there was one in Embrc, which was done a couple of years ago. There was one in Embrc, which was done actually 2 years ago. It was a mix of publication. And 2 from around 15 years ago in Puerto Rico and North Carolina. So we don't know much about the bird diet of these adults. So what this contributes is to show that there is a clear difference how much and what adults eat compared to the chicks. And because these species are specialists, they have been found in previous literature to be a good indicators of climate change. So by tracking what they eat and how much they eat, we know what is present in the field. So that's how we can also see from some of the diet that some cases in the Temperonas, you can see that some adult sandpiper was present in large numbers in the middle of the season, which is not normal for this kind of species. This indicates that the temperature of the sea was better for them, which means usually warmer.
On the way, have a baseline over the adult time.
Yes, as I said, so for this species in particular, this has been done for a couple of years, so they are gathering all the data to get a full trend, see exactly is this really as it is. And there was no many other birds, I said, other marine birds that have this kind of research done to them, at least not by this method or by this method.
Okay, thank you very much.
Thank you.
Time for one more question.
Okay. So first of all, congratulations for this end of the long work to right here. So, and well, I will pay more attention to translation. I will like my colleagues, and I have also the same point and say that I would say that at the beginning it was very nice because I understand the problem. You make a very framework of the path down which species we were working, so that's very nice, particularly for me that related to subjects, so it's great. But at the point you started with data, I was a bit lost sometimes because you were talking about what you really know, but I couldn't follow sometimes what you were talking about. So that's maybe to point it with, to be sure that we look at the same place you are looking, like pointing with the finger where you are talking. That will.
Yeah, I would really like to have like a sticker.
Yeah, that will be
much easier, but
it's not okay. That's just I suggest. And one, for example, in this image, I was for example, I would like to have 2 colors because you are comparing the chick and adults there. but you are using exactly the same color. So at the beginning, I thought it was all for the same. I couldn't make that difference. Now that I have time to read it, I realize like, the first is the cheek and the open part of the other for the first year, and that now I understand the results more easier. So maybe playing with colors would help to do that kind of thing. This is one thing, and I would like to ask you, because we were talking analysis, statistical analysis, which program have you software used for this?
So I use the R primarily to do, and sometimes I use, when for the simpler stuff, I use Excel as well.
Okay.
I compare both to see did I get the same thing, but yeah, mostly I use the R and I use the image J for the analysis of the pictures and authorities.
Okay. But that wasn't mentioned, but and how was it for you to work with R? Was something?
So for example, I am a person that I am not that good with statistics. I'm not gifted regarding numbers in that kind of sense, but I learned much. I developed it. My supervisor told me that I'm better than I think that I am. So I went from knowing quite little to being decent.
Okay.
I managed to do all this analysis on my own.
Okay. And at the beginning, the first graphs you show us were more like counting the numbers of amounts, not really the statistics, the last part of the presentation right now. Okay, perfect. And well, there is a slide where you check, you compare your results with previous results. There is a slide there where you make a summary of what is fit with the previous
Okay, sure.
But you were talking about one column, but I don't really understand the second one. Difference from past.
Similarity to different and differentiates from the literature.
Okay, so yeah, you make it in negative and positive.
Yes.
Oh, okay, it's the same.
This is the X and one. So this matches.
Now I can see that. Okay, that makes sense now. So for me, that's all. Congratulations and how to succeed in the future.
Thank you very much.
Thank you. Seamless transition to the next talk. Our technical team might get slight adjustments to this presentation. Bear with us for 1 moment, please. Deliver of the stick as a
So you can use the laptop, but then you will not be on the camera. You have to stand here. Talking like this, and it's going to be comfortable. Standing here, up here. Okay. Thank you. Very from here. Okay.
all the way from me to you. Welcome.
Yeah.
Very best of potential last week.
Okay, good morning, everyone. My name is Oluwasegun Ike, and I'm going to be presenting my thesis. Topic is identification and characterization of harmful algal bloom cysts from sediment of 10 stations in the Irish coastal waters using multidisciplinary approach by me, and my supervisor is Dr. Luka Merimi at ATU in Galway. Introduction. We all know that when microalgae proliferate in abundance, they are going to form something called blooms. And this condition can result into the production of toxic substances, which can reduce aquatic life, affect biodiversity, and also reduce recreational activities. One thing we should know is that marine shellfish and seafood, such as the bivalves, they accumulate these toxins due to their filter feeding habits. So now, during winter, we try to wear winter jackets so that we are going to be protected. So during the harsh environmental condition, these microalgae in the ocean, they also form something called cysts, which enable them to survive the harsh temperature and also other stress condition in the ocean. So when they form this cyst, they try to settle at the sediment of the ocean. When you see a pregnant woman, what do you think about the future? Of course, a baby. So when we see a cyst in a particular location, it can help us to think of possible algal blooms in a particular area and also the kind of microalgae that's going to be expected in that particular area. In Ireland is very good in production of seafood and they get a lot of annual income from seafood. So previous harmful algal blooms in the Irish coastal waters has led to the development of biomonitoring programs in order to improve the quantity and quality of the seafood production in the country. Now, imagine a layman and a blind man in a particular area. If you inform them about the future war that is going to take place, definitely they are going to run for their life and take proper measures so that they are going to be protected. So the biomonitoring programs, when we are able to detect on time possible, like when we check for the cyst of harmful algae in the Irish coastal water, then it's going to help us to think of what to do in the future so that we are going to improve the quantity and the quality of the seafood production in the country. So my objective, in order to identify and characterize these harmful algae, these cysts in the sediment, we have to separate them from the sediment debris. So I try to use two different methods, the heating and the sonication methods. Then another thing is that I try to check for the variability in the species of these cysts that are present in the sediment with respect to the matrices. What I mean by the matrices is sandy, maybe the sediment is sandy or muddy. So I try to check if there is going to be variability in the species of the cysts that are present in them, because from literature it was known that muddy samples have more cyst count, can retain more cysts than the sandy ones. So I try to use microscopy identification and Sanger sequencing to identify this species. Also, from literature, in this kind of assay, there are two major extraction kits for DNA extraction kits that are used, the Denisi Plant Mini and the Denisi Power Cell Pro kits. So it is very good to compare these two extraction kits that are mostly used in this kind of literature to check for the one that is having more extraction power of the DNA. And also, in terms of the microscopic count among the sediment samples, so I try to quantify the DNA using nanodrop spectrophotometer and the real-time PCR. Materials and methods. So these are the 10 stations in the Irish coastal waters where I took the sample using the Van grab, and also I tried to prevent anoxia condition. I kept the samples at 4 degrees C in the refrigerator. And also I used Wentworth scale to classify the matrices of this sediment, baby sandy or muddy. So I gave them sample code as seen here. So when you see 1SM, it means sample from station 1 and the sediment is sandy and muddy. 2M means station 2 from muddy. 4S means station 4 and it is sandy, and so on. So I tried to weigh 1 gram of sediment and suspended it in 10 ml of autoclave filter seawater. Then I performed the heating and sonication method and checked under the microscope the identification and counting of the cysts in the sediment. Also, as I said earlier, I used two different DNA extraction kits to compare between the two of them, the one that have better extraction power. So I tried to check for the quality and the quantity of the DNA in nanogram per microliter with nanodrop spectrophotometer. Also, I performed PCR and Sanger sequencing and also real-time PCR. So one thing that is important is that I observed that the sandy sediment, it is more better to use heating method to separate them from the sediment debris. And for the muddy samples, it was observed that sonication methods was much more better. So I tried to extract the DNA from the eating treatment for the sandy sediment, and also from the sonication treatment for the muddy samples. As I said earlier, I tried to use the two DNA extraction kits separately. So I used 3 different markers, LSU, ITS, and SSU, which are targeting dinoflagellates in the samples. And I performed PCR and Sanger sequencing. I did some bioinformatic analysis to check for the species present in them. And also I used the 18S marker for the qPCR quantification, which is specific for microalgae in the samples. I used Alexander Menitum culture as the positive control, sterile seawater as the DNA extraction control, and molecular graded water for the known template control. I performed some statistical analysis using the RStudio, and after checking for assumptions such as the skewness and kurtosis and equality of variances and normality test, I was able to decide the particular test that is specific at each point in time. Result and discussion. So these are the 6 images from the no treatment control. What do I mean by no treatment control? So instead of performing eating and sonication methods, I checked directly under the microscope after suspending the sediment to confirm that my eating and sonication methods are actually effective. So from these images, we can see that, of course, I can see cysts, but there are a lot of debris covering them. So I won't be able to identify with morphological characteristics under the microscope. Beautiful, right? So these are the images with the eating and sonication methods, few of them that I put for the sake of this. And as a result of this clarity, I was able to identify them to the genus level. So now, with respect to the distribution of this cyst among the sediment samples, so the positive means present and the negative means absent. So these are the species present in them, Lingunodinium, Diplosplastis, etc. Then I was able to see that with respect to maybe the sediment is sandy or muddy, it doesn't really matter. There is no much variability in terms of the species present in them with respect to the sediment matrices. And one thing is that what can be the justification for this? Dinoflagellate cysts are environmental indicator organisms. So their distribution is influenced by environmental parameters. So during the sampling process, I took environmental parameters such as the salinity, temperature, and I saw that all 10 stations have similar environmental parameters, which might be responsible for no variability in terms of the species present in them. Now, in terms of the average cyst count under the microscope, so this is the microscopic cyst count per 200 microliter of sediment suspension. And this is for the sonication method against the sampling stations. This letter indicates significant differences between and among the sediments. What we observe here, these are the muddy samples, 13M, 1SM, which is sandy and muddy, and we can see that they have higher cyst counts under the microscope than the sandy ones. Also, the same thing with the sonication method. The sandy have the muddy have more or less more average cyst count than the sandy ones. So from the literature, because the cysts have a particular characteristics that is similar to the muddy particles, this might be responsible for high sedimentation rate of the cyst in the muddy sediments than the sandy ones. So there are limitations to the use of microscopy for average cyst count. Is going to there's high risk of error, and it needs high expertise in order not to have false positive and false negative counting. And also, in case of future purpose, it is important to use polarized light and staining techniques to improve the use of microscopy in terms of average cyst count. Also, vegetative culture should be considered in the future so as to confirm the cyst identified to the culture culture in the laboratory after they germinated. This is the result of the PCR, and we can see here that these ones that I highlighted for the 3 markers, the last ones here are the positive control. So the seawater and the non-template control have no amplification, which shows that the work is actually right. Then the 10, 22, and 34 here is the positive control. What do we observe here? For the positive control, there is a single band amplification, and for the sediment samples, we can see multiple bands, which indicates the presence of multiple species inside my samples. For the Sanger sequencing, I have poor quality results with respect to the quality score and the CRL value. So, but nevertheless, I was still able to see the presence of dinoflagellates such as Akashiwo, Alexandrium in my samples. What might be responsible for this poor quality result? This is the melting curve from the qPCR, and it also indicates different amplicons, which shows that there are multiple species in my samples in alignment with the PCR bands that I explained earlier. So in terms of this kind of assay, Sanger sequencing is not the best to be used. So in future, use of high throughput metabarcoding should be used so that we are going to have high quality for the Sanger sequencing so that we can be able to identify the species present in the samples. So now comparing the two extraction kits, the power soil and the plant mini kit. So using the nanodrop spectrophotometer, the plant mini kit have significant higher DNA quantity in terms of nanogram per microliter than the power soil kit in 5 of the samples. Also with the qPCR, we can see that there is significant increase also in DNA quantification with the plant mini kit than the power soil kit in 2 of the samples. What might be responsible for this? From literature, power soil kits have high ability to eliminate inhibitors in the samples. So probably the power soil might have inhibited some DNA molecules, which might be responsible for low concentration of the DNA quantity in the samples that was observed. At the same time, plant mini kits might also give a kind of false positive result due to maybe presence of foreign DNA in the sample. So this is a novel study for future research. So my conclusions. The presence of abscess in these sampling stations indicates the possibility of future blooms in these stations. So there is need for more actions towards the biomonitoring programs. Also, the use of heating and sonication methods actually work for the separation of the sediment debris, which I showed in the images earlier. Also, the use of multidisciplinary approach should be used for this kind of assay because we can see that microscopy gives some kind of results that molecular cannot give. At the same time, molecular gives results that the microscopy cannot give. So combining the multidisciplinary approach helps us to have more reasoning for concluding about our research. And also, based on the matrices, the textures of the sediment, the texture of the sediment does not have effect on the diversity of the species present in the samples, but we can see that it have effect on the average cyst count present in the sample because the muddy samples have higher count than the sediment samples, which I explained earlier. And also, the use of species-specific qPCR and or metabarcoding is recommended for environmental samples like the one I did here instead of using Sanger sequencing. Special thanks to my supervisor, Dr. Luca Merimi, Mr. Dave Clark, my promoter, the PhD student I work with, Erasmus Mundus Scholarship Program, the survey team that helped me to take the sampling. Thank you for listening.
Thank you, well, that's good. And invite questions from the jury, please. Who'd like to go first?
Well, thank you for this so nice and enthusiastic presentation.
Thank you.
Very good. It's very important. Powerful action. Recent program. Congratulations. I understood everything.
Wow, thank you. Thank you.
I think I have no doubts, but I have a question. Have you submitted the thesis already?
Yes.
So these results are
Yes.
okay. Because I read the summary of thesis that you were doing metabarcoding.
No, I suggested metabarcoding. I suggested it as a recommendation. So Sanger sequencing was the one I used because of cost and availability of time. So I used Sanger sequencing, but I was trying to recommend that in this kind of assay, because my Sanger sequencing result was poor, I was only able to analyze few of the samples.
Because I've seen in your results
Yes.
Sanger sequencing.
Yes, I didn't do metabarcoding. I only suggested it for future research.
So I understand. Just didn't understand the reason.
Yeah, now you understood everything, right?
So what say are the main advantages of applying genetics to this your particular case? Because simply from the observation of microscope of your cysts, you were already able to identify the main genera that cause the algae blooms. So it's like what gives genetics
Okay, the importance of
the microscope.
okay, thank you for that. You can see that the microscopy is very difficult to identify these cysts to the species level because even among the same species, there are a lot of diversity morphologically. So microscopy cannot give you that result. So when we do molecular, we are able to identify them to the species level. That is one. And at the same time, the use of this microscopy is prone to error. You know, I'm checking sediment, like trying to check for cysts in the sediment there beyond the microscope. Your eyes sometimes you'll be tired. You can just have false positive or false negative count. So also I was able to confirm that in terms of the count of the cysts in like the presence of the organisms among the sandy and the sediment with the molecular. You know, molecular cannot really lie. It's more accurate than the microscopy, because microscopy, my error is likely personal error. Like the experimenter has more chance of making error because microscopy is going to give you more accurate than the molecular give more accurate result than the microscopy. Exactly. Exactly. Identification and characterization.
Thank you.
Thank you.
Next.
And I would like to join Eva.
Thank you.
I think it's very interesting research, and it's like kind of a step for more to come. I have some questions about the methodology. You have different substances, you have muddy and sandy, and you name one of them, depending on what you have. But when you do the analysis, do you know what you're analyzing each time, or do you blind analysis? Do you know that you are doing sandy or muddy, or
okay, actually, when I sampled and I checked I checked the sediment samples, I saw that there are differences, and I went to literature and I saw that muddy samples have more cyst count. So I was like, okay, let me introduce this into the question. Let me check the variability.
But you know already what you are analyzing. Analysis.
Yes, from like when I wanted to start, I didn't think of that. But after taking the samples, then the equation came up that okay, based on what I'm seeing now, I can definitely include another question because of the variability in the textures.
And the other thing is about the intensifying the heats. All the samples are analyzed on the extraction at the same time. All the samples will be put
at the same time.
The same time. And the other question is that have you done more than one repetition of the result from the sample?
Are you talking about the microscopy or the molecular?
The
okay, for the kits, actually I did it once because of time availability.
Okay.
Yeah, I did it once, so it is important to actually do it again. But actually, that's why I have the positive and the negative control. So actually, give me assurance that what I'm doing is right. Yeah. So with the positive and negative control, I think I'm fine.
So thank you again.
Okay, so I just congratulate you for this nice work. So I have the feeling you have like both different techniques, a lot of techniques in this process, and did you plan it from the beginning to do it, or it was something like, well, let's start and suddenly, well, let's compare with this technique and then and to be sure, or it was really from the beginning?
Okay, actually, what happened was that from the beginning, I have in mind that okay, I'll use microscopy and molecular, but the actual one I want to like the actual molecular I want to do, I didn't really plan it. You understand what I mean? So along the way, I'm seeing the result. Okay, I first started with the microscopy, and when I checked the microscopy result, then I was thinking about the possible molecular analysis that I can do at this particular time that I have, and checking the cost that's available. So I was able to think that, okay, I think I can do this with this time, and I will be able to have a reasonable result.
And well, about the presentation, I would just make a suggestion. When you have the matrix with the positive and negative results at microscopes. Well, I don't know from the last poll, but I can see if it was positive or negative, really small the size. And I would suggest you to do that. Or instead of using positive or negative, better to use 0 and 1. 0 for negative and 1 for positive. It's more easy to see in a matrix the zeros and the ones. And I think it will give you the same information, but easy to see in a first. That's my suggestion for improving a bit the presentation that in the other very much. So, congratulations.
Thank you so much.
Any questions from the audience? Perhaps one or the oh yes. And so just study to establish protocol for good forecasting of establish this as protocol.
Can you speak slowly a little bit?
Do you think you could establish this as a protocol to give warning of forthcoming?
Of course, yes, but this is a little bit like not enough information. You know what I mean? Not really enough information. So if I'm going to be broadcasting for maybe for the country, Ireland, I have to do more. You know what I mean? But at least this is a starting point. Building a room starts in a day. Yeah.
Other questions? Okay.
Gracias.
Next up, we have Samuel.
No, that's all right. Oh, you spelled my name wrong.
Oh, yeah. Wonderful.
Okay, so this slide is set.
Absolutely.
So, okay, so very best of order for this. We're going to hear about the 9-armed octopus beagle.
So welcome everybody. Like we just heard from Ian, today we're going to be going over a behavioral analysis of a 9-armed octopus named Salvador. My name is Sam Su. So before we start, just want to go over some quick anatomical terminology. So when I say anterior and posterior, I'm referring to the front and the back of the individual. And then when we look at actual arm use terminology, going posteriorly on the left side, we have L1, L2, L3, and L4. The same pattern existing on the right side. So octopuses have long been celebrated for their complex body patterning and unique behaviors, with most of these stemming from the use of the highly flexible, usually 8 arms, as shown here in this cleaning behavior done by Salvador. Now, of course, because these arms are used in almost all tasks, they can actually be lost through interactions with predators. But octopuses have the unique ability to regenerate said arms, as shown here in our study's individual, who lost arms R1, R2, R3, and the tip of L1, shown here in purple. Now, although R2, R3, and L1 regenerated normally, occasionally during the process of regeneration, the arm can actually split, and in place of one arm, 2 can grow, a term called bifurcation. And so this is actually what occurred with our individual, and we have the terminology here with R1A and R1B, so providing 9 arms instead of 8. Now, although this has been studied and there have been records, almost all individuals actually noted or recorded are dead or previously found by fishermen. So this is the first in which we found them alive. And now, because although the arms are highly multifunctional, like we said, they can be used in almost every behavior, there are specific patterns that have been shown. So for anterior, posterior, and the R3 arm. And now I'm going to make specific notice of the R3 arm. So this is actually in male octopus. This is the hectocotyledged arm, we call it. So their penis. So it's always the R3 arm on the octopuses. And so this has been shown, for example, in decreased risky usage. Because obviously you don't want to be sticking your reproductive organ in holes and whatnot. So there's also been shown to have specialization that exists on a bilaterally symmetrical axis. Now, although these studies have been shown, there is a serious lack of environmental relevance. So all of these have been done in laboratory settings, and thus we miss a little bit of the actual environmental relevance. So to help fill some of these gaps, we here present the first ever video analysis and any ever videos of octopuses with abnormal, live octopuses or any cephalopod with abnormal abnormalities. So here we employ novel quantification methods for video analysis, corroborate some of those previous patterns, and of course describe and categorize arm usage and its change over time. So moving back to the study subject, here we see Salvador in the most common position with the bifurcated arms curled up, resembling that almost of a mustache. And here we can see that Salvador is a male, as we see the hectocotyledged arm. So if we look at the tip right here, versus the tip of a normal non-hectocotyledged arm. So to actually obtain data, 24 videos collected from citizen science were used, which spanned, of course, of 147 days, which approximated around 1,900,000 frames of video that were analyzed, which took quite an arduous process that took around 2.5 months. From these, watching the videos a couple times each, a component of catalog was made. So every time a arm was moved, the head dropped, a sucker was expanded, it was made note. But obviously, because this is a massive, massive data set, to streamline the behaviors of each of those combinations of these components and their associated categories were put into an ethogram, which was then put into the behavior recording software Boris. Now, Boris allows you, a little bit difficult to see here, but Boris allows you to assign specific keys. So this is W, and this is a behavior called web-over. Allows you to assign specific keys to specific behaviors so that when you're watching them on the screen, you're able to say, for example, we have arms curled as noted right here. You're able to press that key, and you're able to record the behavior. And now behaviors are recorded as what we call events, which is the start and the finish of a concrete behavior. To look at arm usage, we used something called modifiers. So after you click your behavior, this screen would pop up and you'd be able to see exactly which arms were used. Now from this, to test the arm and behavioral associations, we conducted a redundancy analysis, RDA, whose outputs are shown here in this tripod. So the actual arms are shown in red, while the behaviors are shown in black. Now, the actual proximity of each of these terms to each other signifies some sort of association. And further, each of the dots are actually events, so these events that we talked about earlier, but they are aggregated by behavioral category. Now, some patterns we can see from this. So if we look at red here, which are all the locomotion behaviors, namely bipedal walk and crawl, they are more associated with these posterior arms. And for example, we take a look at explore and foraging, which are blue and light blue. Light blue isn't really coming through, but they are more associated with anterior arms. Additionally, we see pink, which is feeding, is most associated with these two bifurcated arms and L1 as well. So the very first two arms. And then the last pattern I want to draw your attention to is the grouping of anterior and posterior arms. So these arms are actually used in similar behaviors. However, we do have one outlier, which we will talk about later. So in this video, we can see some of these patterns we talked about, the use of the back two arms for walking, the use of the arms throwing over, what we call a web over. And then if you see, watch right here, the two bifurcated arms are actually tucked underneath the body to help manipulate and feed. Now, the same type of analysis was done on the same data, but now instead of grouped in by all our different behavior categories, we changed the grouping to have risky and safe behaviors, with risky behaviors being those that exist far away from the body and safe being those that exist close to the body. So for example, curling. We also combined R1A and R1B into just R1, in part to help allow future comparisons for studies. Here we can see that although risky behaviors are kind of a lot more distributed throughout all the arms, safer behaviors are more associated with the arms that are previously lost. So when watching videos, we also noticed some previously undescribed behaviors, which was very fun. So the normal behavior A you see above is called a retroflex, which is a defensive behavior octopuses use when they're in their dens. The arms go above the head, almost as a protection. Now in Salvador, we see down here in B and C, instead of the arms actually existing behind the head, a little bit difficult to see, but they're actually crossed in front of the face, almost as if it was a shield. Additionally, if you remember that term we said earlier, web over, so normally the arms are thrown and the webbing, which is this part in between the two arms, is extended. But here we see Salvador puts the arms underneath, again, that specialized usage, and then crosses the two posterior arms. So a kind of a new behavior that has not been seen before. When looking at usage over time, we see here, a little bit difficult to see, but if you look right here, the two bifurcated arms, this is from the first video, are always curled. So they're not used in walking, they're not used in exploratory reaches, they're not used in any behaviors. While if you look at the usage of all the other arms, they're being used for almost everything, and including the R3 arm. Now, if we compare this with usage later on, as seen here, we can note that the bifurcated arms, you can see they're now being used in a lot more activities. And again, you see right there, the R3 arm is now being curled, so it's not being used as much. But all other arms remain their high usage. Now, this was confirmed through the use of linear litter models. So here we see for risky behaviors that the bifurcated arms increase in usage over time, while almost all other arms decrease in their usage. So when we're interpreting these results, first we see that there was a divergence from the bilateral specialization. So usually, again, R1 and L1 have the similar functions. But if we see here that actually L1 existed a little bit closer to R1B. So although they are not as close proximally, they actually serve similar functions. And then again, we can also see an increased specialization of these two bifurcated arms for these feeding behaviors, specifically manipulate and tucked under. We can also use these studies to confirm previous laboratory results for those patterns of behavior, locomotion and anterior or posterior and anterior arms. But then again, there was that divergence of the hectocaudalized arm. So we thought this was due to the fact that because those two small bifurcated arms were not used, there was a compensation by an arm that is most commonly not used in these behaviors to help allow to, for example, acclimatize food or in foraging and feeding. So it kind of shows a degree of individual flexibility and cognitive decision making, as shown in this video. These results also allude to a degree of learning and problem solving, which has probably previously been shown in these species. Learning in the sense that these arms increase in their usage over time, which allowed things such as higher prey accumulation. And then problem solving in the degree that, as we see here, there was a small gap that existed because of the smaller size of the bifurcated arms, which in turn led to problems with food loss if you just look at that same area, you can see that, unfortunately, food was lost through this hole. So maybe there was some awareness of this, and that's why we see this compensation in, for example, the crossing over of the arms in those previously well-over tactics. These results also help us further support the presence of memory, and specifically post-trauma associated memory in Octopus vulgaris specifically, and the genus as a whole. So in a study done by Cook in 2021, they showed that a nitrous stimulant was administered to octopuses in a specific area, and then they registered pain usage, and then the octopus was actually shown to avoid said area on a second administration. So understanding that this area is associated with pain, I don't want to be in this area. Further, they showed that there was wound grooming behaviors. So for example, if you run into a door frame and hit your head, you're rubbing your head. So something similar to that. And then that actually rubbing corresponded to the degree of pain administered. So longer groom behavior for higher pain increases. So in Salvador, we see that the usage of previously fully lost arms in safer behaviors. So again, possibly alluding to the fact that these arms were previously lost. There's an understanding that I don't want to use these same arms in these risky behaviors, so I will thus keep them close to my body as compared to, for example, these non-previously lost arms. So, of course, it is the conservation and management of species is only truly possible if you have a grasp, a good understanding of ecology, life history, and behaviors of said species. And further, as most species are in a decline, the cephalopods have actually been showing to proliferate in their populations due to things such as RNA editing or their short lifespans and behavioral elasticity, as shown in some of these studies. So understanding this proliferation through behavioral studies such as this can allow us to not only have better self-conserve cephalopods, but also possibly all marine species. Finally, although this study was somewhat complete, it does serve as a baseline for further research. Now, it is difficult because there are not many registered 9-armed octopuses in the world, but the same things can be done, for example, running a meta-analysis for 8-armed octopuses, because, of course, more individuals are needed, as this was only done on one individual. And further, as octopuses have been shown to have unique personalities, what one individual will do will not be the same as another. There are also some really fun neurobiological implications of this, and we are in talk with a couple of different neurobiologists in the field to try to help answer some of these questions. So some would be, as octopuses have 500 million neurons, roughly around the same as a dog, they're distributed all throughout their entire body. So what happens when an arm is lost, for example? Are those neurons lost with the arm? Are they redistributed throughout the body? And when that arm regrows, are the neurons regrown properly into the arm? Are they regrown in correlation to size? Lots of fun questions that will remain just that for now. Questions? So thank you first and foremost to the late Andrew Packer and Eric Hochberg, who were pioneers in this field and inspired many cephalopod biologists such as myself. To my supervisors, Jorge and Miguel, of course, thank you for everything. And to our citizen scientists and cameraman, Hide, you are inspiring, and your dedication to your craft created this entire project. So thank you very much. Mm-hmm.
Thank you very much, Sam. So I'd like to take some questions from the jury. Would like to go first?
Okay, congratulations. Very nice talk and really very concrete. I would like to that's good. I'm enthusiastic when talking, but very nice because I could understand all. So congratulations first for this very nice work. I was just when you were talking, I was just writing, making questions, like to understand what you have done. One of the questions I have is, how did you start this web? How was the first question you made to see, okay, I'm going to hold the octopus for hours to see how they move? And for me, it's like, wow, I can imagine how the idea comes.
Truly. So all of this, although I wish I was in the water with this octopus, all of this was recorded by our citizen scientists, who then were able to contact us through Dive Camp, which is a program in the Balearic Islands. And more or less, I think he found this individual and noted a specific anatomical morphology or morphological abnormality, and contacted us, and we were able to kind of just pressure and say we would love to have more videos of this. And then naturally, as the arms are used in almost all activities, the speculation that it would possibly change said behaviors was kind of in the back of our minds. We wanted to see how it would change over time, and just overall just kind of explain what has not been explained before.
Okay, and as I understood, it's sugar. It's like this is the first one that you find alive. It's bifurcation. And it's just you were lucky, or there is a relation between that you suffer this bifurcation, then you have more probability to die and find this kind of.
That is also possible, but to answer the first part of your question first, I think there was probably a degree of luck as well, because although again a lot of them have been found, and there was actually not just bifurcation exists, but polyfurcation, so octopuses can regenerate all arms, such as this 90-armed individual. But again, most of them have been found dead, the coffin fishermen. So I think we were very
well. And there were this kind of uncontrolled growth.
Exactly.
Finally.
So yeah, so it was possibly a mutation in the Hox gene of the homobox gene group, although it's very understudied.
And did you analyze the behavior of arms, and how do you compare it with, say, like as expected and only change for the bifurcation, or is changing also from what you expect from those that are
with normal arms? Yeah, so that was in part what the redundancy analysis allowed us to do. It allowed us to actually look at these specific patterns. And our great next step, I think, would be to do the same type of analysis, but maybe using AI, because to do close to 2 million frames of analysis is intense. So to use a automated system that could then replicate this, but for a 8-armed individual, so we can see the same patterns and compare and contrast them to see what the bifurcated arm actually does specifically.
And I have one last question that was already on future research, that is how it affects this bifurcation in the nervous system, and it's a kind of relation and it's modulated, but you have it already. So I hope you can find answers to that question.
Yeah, of course.
It's interesting to see how it's affected, because also we probably give more information about how the nervous system works, not only for them,
but for humans as well.
So thank you very much, and congratulations.
Thank you. Yeah.
Wow. The persistence of memory. This is a title.
And it alludes to the name of the individual as well. Of course, after.
Absolutely, I like it very, very much.
Thank you.
And it's not you. I understand all the presentation for me. Spoiler. My question, perhaps I missed it's too much information. What would years of payment, and is a single octopus for many of the persons.
So yeah, so the videos were obtained via filming from a citizen scientist who provided them with us, and they are all of one individual.
Why?
Which is why the need for further studies would greatly benefit this.
Because
Some
the method was simply observational and was non-intrusive.
Exactly. So it was all from video data that were previously taken.
Perfect. That was my answer.
Yeah.
As you know, if the sanitarium during the complement research code.
Of course.
But now, I'm sure you didn't start after the video.
No, it was truly incredible to see the comfortability and I think which is why it's quite unique data set, because the octopus after a couple of days got quite familiar with the photographer, videographer, citizen scientist. And so the actual behaviors we're watching aren't kind of adjusted in any way, shape, or form. At least we think, of course. But yeah. No, very, 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 very comfortable.
That's exactly. You have explanation for the like the behaviors or still sitting down with that. I know you explained how.
Yeah, I think to a degree it's just streamlining your own efficiency with maybe your daily life. So, for example, Salvador finds it easier, or the individual finds it easier to cross the arms like this, as opposed to exert the energy to put them behind. Kind of like some of us brush our teeth before we wash our face. Some of us wash our face before we brush our teeth. It's a personal preference.
So, in principle, it's very diversity of behaviors, point that sort of intelligence and some way.
I think, yeah,
As
so.
proposed
Because of course all this has been very previously shown and quite well studied as well.
And we mentioned some
Yes, so the bifurcation, it is, again, although it is understudied, the suggestion now is that it is a mutation in the Hox group of homobox genes. So, right, these genes allow or help show the regeneration of limbs and help allow on an anterior to posterior axis. So a mutation in this could possibly result in the splitting of arms and a change in where anterior to posteriorly they grow, develop. So,
Thank you very much. Congratulations.
Thank you.
More questions?
Just a couple of thank you for the talk. And I have this. And during all this time you were for so that they does the bifurcated stay same size all the time or does it grow?
So yeah, so I think the reason that the usage increased is because as time progressed, the arms increased as well. So it could be degree of, although it might not be decision making, it could be, okay, these arms are very small, so it's not beneficial to me to use them in these activities that exist far away from my body when I have a super long arm right next to it. So that would be, I think that'd be a wonderful next line of research to kind of take into consideration length over time and see how that changes with the usage.
And the last thing, because I have made it. The other thing is, I saw that when you show the same behavior, there's difference between the right side and side. That's normally.
So octopuses have been shown to have a side preference that usually follows an eye preference as well. So, for example, in a study, they showed in the recollection of food. They created a T-maze, which is just this big T-shaped PVC tubing, and octopuses, depending on the individual, use a specific side or preference.
So it's like our left,
Exactly.
right
So
hand.
it's kind of just like us, left or right-handed.
And the last one.
Mhm.
Did you find, for instance, self behavior the animal that has the
Mm.
will have more say behaviors more than animals?
I did not compare it to any other 8-armed octopuses, but that would be a very interesting combination to do down the road. Kind of either via meta-analysis or because it's possible that again, it maybe is a little bit more cautious in everyday life as compared to somebody that is never had an incident or an injury. Yeah, there's a lot of wonderful things that can happen after this.
Thank you, David.
Thank you.
Any questions from the audience? Thank you.
Very physiological arrangements.
Yeah. So morphologically, no, they were quite similar. But in their usage, we do show here that R1A was actually a little bit more associated to. So, for example, this is the most safe behavior that this individual showed, just the curling of the arms, so keep it tight to the body as possible. So R1A actually showed to be a little bit used to increase safer activities versus R1B, which is a little bit more associated to approach, to pouncing, a little bit to exploratory reach. So super interesting thing that I was thinking about was, right, there's a preference that exists within individuals, but maybe there's a further preference that exists within these two black-eared arms. So like a specialization.
No questions on that? Oh, one here in the left. Okay.
I was curious for that curious person that's filming water showing how she was sure that it was the same time. So to observe and everything and how is that?
So besides the extra two arms, octopuses do have specific patterning. Of course, changes over time, changes with behaviors, but kind of specific markings. But I think the big one was 2 arms. There's not a whole lot of octopuses. But again, it was in part very, very informed. He's been diving in this area all the time. So he knows where the ends of each of the species are, the individuals live, where they move. So yeah, very lucky to have.
Last one, so okay, last two. So.
This is a good question. I was mentioning the with already. Why is that?
Well, if you remember that initial position that I said when we talked about the hectocardialized arm, so right, this is the most common position that Salvador assumed. And if you're asking me, it kind of looks like a mustache. And Spanish octopus, eccentric person, it works. It works.
Moving on.
Yeah.
It's like the length quite long, right?
Mhm.
And how
Yes.
did you film? Like, did you install a camera or
So
for the very long time?
So it was very long dives. And yes, so video ranged from the longest was an hour and 56 minutes, and the shortest was 13 minutes. But so just follow the individual throughout different time periods. There are shifts in and out. So probably leaving the water to maybe recharge a tank or to recharge a camera. But all of it wasn't usable. So around, I think, a mean of 85% of the videos were actually usable. But of course, octopus enters an area where the arms can't be properly seen, so I can't properly do analysis. But
Sam through it all.
I'm ready for it. More if you need it. Yeah?
Session Manuel.
How do I put it on slides mode? I don't know how to put it on slides mode. What? Because it's a PDF. Then I should be able to put it on slides mode. Just over. Yeah. Yeah. What's this? Thank you.
Okay, so Manuel, after your 5
Thank you. Hello everyone, I'm Emmanuel Jarin. I'm happy to talk about my master's thesis that I've done in the biological station of Roscoff under the supervision of Anne-Claire Pascal Riera, and Nathalie Simon. And it was about the impact of the viral infection on the metabolism of the nanodiatom Medullabrus comicus, so way smaller than octopuses. First things first, why do we want to study phytoplankton? Because phytoplankton are primary producers and they're going to transform the atmospheric CO2 into organic matter, making it available as a source of nourishment for every other upper trophic level in the sea. And through this, they're important in biogeochemical cycle and especially carbon. And thanks to their metabolism and photosynthesis, they're able to do that. There are several factors that are going to regulate the metabolism of phytoplankton. First, there's abiotic factor, for example, light, nutrient availability, or temperature. And we are able to measure the impact of these abiotic factors on the metabolism by looking at growth rate or chlorophyllite content or nutrient incorporation inside our phytoplankton cells. On the other hand, there's also biotic factors that are going to regulate our phytoplankton metabolism, such as viruses. Viruses are going to infect that host, and they're going to pull out their genes inside the host and change the machinery of the host to allocate it for viral production. And through that, they're going to lead to the lysis of the host. The lysis is the moment in which the host is going to die, and he's going to release the viral progeny and the cellular content back to the environment. Mostly, lysis parts have been studied so far due to its evident impact on biogeochemical cycle. And we wanted to focus on the moment in between the infection and before that lysis, because during that moment, there is a reprogramming of the host's metabolism for the production of viral progeny. These metabolic changes happen during the infection, and they have been studied in cyanobacteria so far. And in cyanobacteria, they've underlined that during the infection, the virus could inhibit carbon fixation or upregulated nitrogen uptakes, because nitrogen is more needed for viral production than carbon, for example. And we wanted to follow that on another host model virus. And we've decided to focus on diatom, because diatoms are an emblematic group of phytoplankton, and they're responsible for 40% of primary production. And in our virus host system, we focused on Medullabrus comicus. Here you have the proportion of reads of diatoms over time in Roscoff water, so where my study took place. And you can see that Medullabrus comicus is dominant in that region and blooming during winter. And she's a nanodiatom, so ranging from 2 to 5 micrometer, which is very small. And for these reasons, she has been very understudied compared to bigger diatoms. So we focused on that virus host system, Mediolabrus comicus, and our associated RNA virus. And we wanted to answer one question, which was, does viral infection alter Mediolabrus comicus metabolism and its biogeochemical role? You have here a little picture of our nanodiatom. To answer that question, we focused on two research hypotheses. The first one was, does infection affect host growth and physiology? And the second one was to know if infection affects carbon and nitrogen incorporation in our phytoplankton. To answer these hypotheses, we followed cultures of Mediolabrus comicus and our virus in extended conditions, so without any bacteria, and we incubated them at 18 degrees with a day and night cycle into a silica media. Here you have a healthy host, and here is an infected one, which is transparent because of the release of its contents. To follow this culture over time, we've measured several parameters. We've measured diatom's abundance and cellular properties such as size, granulometry, and chlorophyll A content. To do so, we use a flow cytometer. We've also followed RNA virus abundances using dilution by extension, which is a biomolecular method. We followed photosynthesis system health status of our host, and especially by taking a look at protein complex 2, which is important for photosynthesis. And to do so, we used a spectropluorimeter. And finally, we took a look at incorporation of stable isotopes C13 and nitrogen-15 in our host. And to do so, we used an elemental analyzer coupled with an isotopic ratio mass spectrometer. You're never going to hear that again in your life. You're good. So we studied a new method on a new host virus system. And to do so, we needed previous knowledge on our model. So we conducted 4 pilot experiments following the entire life cycle and including lysis. And to do that, we had 4 culture conditions. The first one is the host only. Then it's the host enriched with stable isotope C13 and nitrogen-15. Then it's our infected host and infected enriched host with stable isotopes again. So I'm going to show first the results of the pilot experiment to make it more clear for you after. Here you have the abundance of diatoms in cell per milliliter over time. And you can see that after 96 hours, we have a collapse of these abundances and then a return back to normal. So, sorry, in blue, it's our host, and in red, it's our infected host. For some reason, the rest is not showing, but... And so after 96 hours, you have a collapse of the abundances, and this is when lysis happens of the host. So I want to focus on that part. Then we took a look on our cellular properties, again with in blue the host only and in red the infected host. And before 96 hours, in terms of size, we have a slight increase after 1 day of infection. It was the same for chlorophyll A content. We had, after 1 day of infection, a slight increase in chlorophyll A content in our infected cells. And granulometry didn't show any difference. Finally, during our pilot experiments, we added the stable isotope enrichment. Here it's the solid lines. In red, it's infected, and in blue, it's the host only. And we wanted to see if our enrichment had an impact over our culture behaviors. But it was the same if it was enriched or not, so we were happy about that. After that, we set up a final experiment to clearly monitor carbon and nitrogen fluxes in our cultures. Our aim was to do it on 5 days to monitor the beginning of the lysis, but it actually happened over 3 days because we had to restart the experiment due to bacterial contamination in our viruses. So this final experiment was set up like this. It's quite tricky, but you have 2 series because we were afraid that the diatom would eat the entire enrichments that we put. So we did 2 spikes. So there's the 4 conditions in triplicates, and it was enriched at T0 in series B and followed until 40 hours. And then we did the same with series B, and we spiked the enrichment at 40 hours and then followed it until 72 hours. Anyway, these all cultures were followed every 8 hours, and we measured abundances of diatoms of bacteria, membrane integrity, photosynthetic efficiency, carbon and nitrogen incorporation, and once a day we followed the viruses. We had different results in terms of abundance and cellular properties that you can see here, abundances, and here it's a proxy of size of diatoms over time. There's a high variability in between our cultures, and there were no differences in between infected and non-infected cultures. Probably because over 72 hours, since we are not looking at the lysis, it's too early to see differences. Despite this result, we saw that viruses over time were released after 48 hours, meaning that we had a production of viruses in our cultures. And this was concomitant with the fact that the cell of the host lost some membrane integrity and so were compromised after 48 hours and 72 hours of samplings. In terms of carbon and nitrogen incorporation, I wish I could be more positive. We had mild changes. So here is the carbon over time, and here is the nitrogen over time. And our hosts only are in blue. So you can see that after 48 hours, there seems to be mild changes, and we seem to have like infected cultures that incorporate a bit less than the controls, but it was non-significant. And when you took a look at the photophysiology analysis and so on, the photosystem generally, there's no difference of efficiency in between infected and non-infected cultures. So to summarize all that, we have no changes in abundances over 72 hours, neither in cellular properties and neither in carbon and nitrogen incorporation during our final experiments. This was not the same in our pilot experiment because in terms of abundances and cellular properties, we had differences. So I think what happened is that since we restart our final experiment, we infected a host that was in late exponential phases, and the viral infection will have less impact or less quickly on a late infected host. So if we hadn't restarted, that might have worked better on the final experiment. In terms of carbon and nitrogen, we didn't have any significant difference neither. Maybe diatoms are of different behavior than cyanobacteria that we've seen before, and maybe they are more resistant and there's no difference in metabolism in between infected and non-infected cultures. And it's more likely due to a high variability between our replicates, because we worked with true replicates, so with different flasks, and with 2 series, which makes a lot of variability, and these kind of measurements need to be probably more precise. So to optimize this final experiment, we had to remove the bacteria, and to do so, maybe a chemical treatment would be needed. And also, I think this double spike idea of doing a series A and B would not work, and it would be better to have a continuous inflow of enrichment in our cultures. So to go back to our research hypothesis, infection affects outbursts and physiology. I would say yes, because we still had some differences in our pilot experiments. And in terms of carbon and nitrogen incorporation, with optimization of our final experiment, maybe we could see differences in the future. To finish on a positive note, I still think this master's thesis developed an innovative methods to follow metabolism of infected nanodiatoms during that study. I would like to thank all the people of Roscoff, especially my 3 supervisors and the people that helped me during my final experiment, because it was tough. Thank you for your attention.
Thank you very much, Manuel. Thank you very much. So, have I questions from members of the jury, please?
Yeah. Thank you very much for your presentation. And I really enjoy it. It was somehow. So that's very nice. And I have forgotten since I'm almost I also think that the lack of significance makes a sense of that's how we have
Yeah.
make them very. So what would you think?
It's hard to say because so working with true replicates is maybe to like maybe it would have been better to sample in the same flask and have like a bigger culture, but having bigger volume in extended condition is also tough because like materially in terms of material it's hard to sterilize bottle of big volumes and under the hood it doesn't work. So I don't exactly know how we can make it work with this variability. Yeah, increasing the number of replicates would be best. Sorry.
And the other thing is just first time experiment. One, you have like a certain number of cells
Yeah.
after 7 hours.
96, but
96 hours, sorry. First, but having
yeah, it's strange. This one and compared to this one. Yeah, so it's hard to explain because we don't have more microscopy to help us here. So it's only flow cytometry. But there's studies that have shown that during infection, the biovolume can increase, and also the volumes of thylakoids and chlorophyll A content. So maybe that could be an explanation, or maybe the release of the contents of the cells. I can't be sure here.
Thank you very much for the presentation. And there, always very good and speak.
Thank you.
For me, negative results, I think the results weren't
Yeah.
expected, are also results, and it can be very results. You only have to explain that why the hypothesis failed or was not supported from good data in that specific experiment. Thirdly, well, first, there is of action action on this for wider. But you think it's true? I mean, or is perhaps the experiment itself, or the design of the condition?
I'm not sure I understood your question. You're asking if. Can you repeat? I'm sorry.
Expected an effect, a negative effect of the viral infection.
Well, it doesn't have to be negative all the time. It can also increase fixations. Everything is possible actually. It can enhance sexual reproduction. It can do so many different actions like COVID for us. It had so many different symptoms. It's the same for them. But yeah, we're expecting to see the same pattern as for cyanobacteria because it was the easiest hypothesis it was the only study we could refer to. But yeah, it might be different. Maybe nanodiatoms are behaving very differently and they're more resistant. these cultures were actually very resistant. They were easy to grow. So yeah, yeah, it's positive sometimes.
Primary infection at risk viruses.
Mhm.
This is good. Actually, how frequent are those viral infections?
So half of the mortality of phytoplankton is due to viruses. And in terms of infection in the natural samples, we still don't know exactly what portion of phytoplankton is infected, but it's in development because it's very new. They're managed now to know how much percentage of mortality due to infection. It's still not exactly the number of infected cell in a natural sampling.
So the plankton height has been defined based on studies or
experimental studies.
So this take so but it's the first time this experiment combining virus and this diet of growth.
Yeah.
So I didn't know what to expect.
No, I worked in the blindly. But that was
Many days I it's good.
Mhm.
Because my first one that I put first statistics was in this section alternate way than the primary class, but the answer was the results. But
it's hard to say. Depends on the phytoplankton. I think.
thank you.
So congratulations, first of all. It's a nice talk also. I'm not sure I really get this picture. So we were trying to understand how the virus is affecting the case, and in particular, we were trying to see some factors to lysis and to control this virus infection or not. Why we were focusing on this carbon network element, this analysis, to be able to somehow predict what is going to happen before the lysis and then some kind of action, or just
the main aim is to compare fixation of carbon and nitrogen in infected and non-infected cells to see if when they're infected they fix, for example, less carbon, and have an idea of the general impact of viruses in the sea of phytoplankton and how they impact viruses this carbon cycle. Is that more?
Yeah. Okay, but then if you are not focusing on how the virus is affecting from mortality point of the plankton, make decisions to help. I'm thinking in infection models where you are following the virus infection in a population, try to establish or to understand how the infection is spread in the population and then to cut the infection. But that's not the goal.
No.
But that's it. Okay.
Yeah, that's
yeah.
It was just to see when lysis occurred.
Okay.
Because we didn't know.
Yeah, I was just thinking on these infections models where you have this kind of population and trying to see how it can be connected. And also, I was going to ask you somehow find some mathematical models that can support also this analysis and to compare with simulations.
This is actually, there's labs that are working on that. I know some people also working on temperature impact over viral cycle and trying to see by modeling. But it's good to have experimental too.
Yeah, of course. It's like you have to together,
really,
working parallel in that sense.
But yeah, there's some modelization.
But you didn't try to
no.
compare.
That was
just also for me, it's a prayer to see that growth and increase academic growth.
Yeah.
So I did you try to, well, maybe it's not possible to go a bit farther on time, or?
No, I haven't. But I agree that would have been good. It's just in terms of, I haven't done it after.
Okay, yeah. So that's where my doubts. So thank you very much.
Sorry.
So before you
can you speak louder because I don't hear anything?
Yeah. Before you control the data and the bias to check for life, do you treat the data or do you mention about bacteria?
That's a wait.
Okay. And the data, before you control them, they have the bias. Do you do any treatment that? Do you use physics? Yeah.
Yeah, so it's very okay. It's hard to explain, but so I have my dietome, and she was extended because she was treated with antibiotics for anemia, so she had no bacteria. But the problem was my virus that I had it into my dietome to infect it, because this virus was filtered several times on the 0.0 mesh, but I still had somehow the bacteria on it. And it was impossible to get rid of it.
Let's think two apps and interaction.
Yeah, they could. That's a possibility. That's why I don't want them to like just one system, one dietary and one virus. So you have to have
Question one.
Does viral infection result lysis and dietary?
No, there's different cycles. Some of them are symbiotic, but most of the time, after, for example, a stressor, the diatome will still go to death. But they can live together for a time.
Okay, so folks, I think that will draw the session to conclusion. This morning, this afternoon, we heard from Robert, who told us about a new method of dietary analysis for sandwich terms, which gave information on dietary differences between adults and chicks over the space of time and see. Although Aspen investigated methods, performance about what is hiding in the sediment of fire, jeopardize food safety and security. Sam told us about Salvador, who once bitten, twice shy, and perhaps involved compensating for damage. We developed different patterns of RNUs, supporting models of a highly evolved species, and Manuel told us about the viral infections of narrow diatoms, which led to exposure in this case findings which she explained. But the study developed little methods of further investigation. I'd like to thank the speakers, I'd like to thank the jury, Garcia, and I'd like to thank my teacher for support.